Okay, I think everybody is back. So maybe I'll start by asking the instructors from your rooms, were there any specific gene models that you thought were interesting and are worth looking at? I can share my screen and we can put them up there. Or if you think somebody in your group would like to discuss their gene model, that's obviously also totally fine.
Hey, Mar, in group 4, we had an interesting gene. I wonder if you could actually take a look and maybe see what your opinion would be in terms of the RNA-seq. It was the third, I believe, the last one, the 15. Yeah, that one.
Okay.
There was some sort of interesting intron mapping from the unannotated tracks on the right side of the gene that did not match, obviously, anything that we have had in the gene model. And if you activate different types of the RNA-seq data from some, it almost as the gene was actually a lot larger than what we show in our gene model. So if you could offer perhaps a summary of that would be fantastic.
Sure. So let's take a look at it. So I went ahead and copied the gene ID, put it in the gene finder right here in the box, it loaded the gene model. It typically, when it if you notice probably in the genome browser, it tries to zoom into sort of the gene region, so I'll always have to zoom out a little bit. And then the second thing I did is I pinned my gene annotation gene model track to the top, and I also pinned the introns to the top. So this way now, when actually, maybe this is not a good idea because there are many introns here. So I'm going to unpin. Do that again. I will unpin the introns. Or actually, what we can do is let's make this collapsed. Okay. So what this allows me to do is to still see the introns right here. They're collapsed, so there are multiple introns overlapping here. We can always expand this again and take a look at it in more detail. Okay, but now I can scroll down and see the RNA sequence data. So I'll explain what tracks I have on. So one track that we discussed in our group, which I don't think is in the exercise, but there's a track for transcription start sites in ToxoDB. And so that allows you to see, in this particular experiment, they sequenced specifically the transcription start sites based on the technology they used. They were able to pull down full transcripts and then sequence their ends, and then mark exactly where those locations are. Obviously, there are false positives in there and negatives, but they're kind of the idea. I think hopefully you're appreciating with all of this is the more data you have, the more data you can put together and sort of come up with a summary conclusion based on different data types and potentially different data from different groups. The other tracks we turned on were these ChIP and ChIP-seq tracks. So I actually went in and I found a ChIP track for histone 3 K4 methyl 3, and then I found the ChIP-seq track for the same histone mark. Okay, and again the same thing for the histone 3 K4 methyl 1. I found the track for ChIP and ChIP-seq, and I was telling my group that even if you don't know anything about what these histones, what do they mark in the genome, hopefully you can quickly appreciate that, well, this histone 3 K4 methyl 3 seems to have peaks in between genes. Maybe it has to do something with the start of the genes, and you can sort of move around and see where those peaks are, and they seem to always coincide with the beginning of a gene. So that's one observation. The second observation is that the ChIP data and the ChIP-seq data correspond to each other. They're pretty consistent with each other. And that's pretty cool to me because these are experiments that were done probably 10 years apart using two different technologies from two different labs, right? And they're giving you basically the same answer, which is really gives you great confidence about the reproducibility of experiments. And then looking at the second track, you will see that you kind of almost have the opposite view. It's lower in the places where the promoters are maybe, or the beginnings of genes are, but you start seeing some expression or some more reads mapping to where the genes are. So if I had to make a conclusion, I would conclude that that second one, the histone 3 K4 methyl 1, is marking the transcriptional units. And that turns out to be exactly what these do, but it's kind of nice to see that you can without even knowing anything about them, you can start making some conclusions about what they potentially do. And then the other thing I have is I've turned on the RNA sequence data, and I turned on the RNA sequence data from this cat stages, and actually this experiment has cat stages, and then it has also the tachyzoid and bradyzoid stages or cyst stages from Toxoplasma. They're blue and red, and so this is because it's a strand-specific experiment. It's a strand-aware experiment. So the experiment is done in a way where each strand of the transcripts coming from each strands of your DNA are you're able to mark them and follow them through the entire experiment. So in the end, when you get your reads, you're able to tell, well, these reads came from the forward strand and these reads came from the reverse strand. And that's pretty cool, because now instead of not knowing if a bunch of reads, like for example, if this was a non-stranded experiment, you'll see something like this blue track here, and you won't know, well, is this peak here and this peak, do they belong to each other or are they separate genes? So now seeing the RNA-seq data, you will see, so you'll notice that there are actually a couple of things going on here. There's also red tracks here that potentially go in with this red gene going in the opposite direction. I didn't do a good job explaining this, but the bottom line is it helps you not only see the expression of the gene, but also the orientation of the gene, and also separate between potentially ambiguous regions where gene models may overlap. Additional tracks that I turned on are the community annotation. It's always nice to see that. We also have nanopore, long-read nanopore data. And so each one of these technically signifies an RNA species. It obviously has its issues. And then in the end, we were talking about this alternative gene models based on this program called Craig. It's really not heavily used. It's actually only available in ToxoDB, but that's an RNA-seq or transcriptomics aware gene finder, which tries to predict gene models using RNA-seq data and ESD evidence data and so forth. So it's kind of okay to look at as an additional source of evidence. Okay, so looking at this gene region here, so what are some conclusions that you can make? So one conclusion, just quickly, just looking at the RNA-seq data, you can see that there's almost no expression in the cat stages, but then it goes up there's expression in the tachyzoid and bradyzoid stages. So one conclusion could be that this is maybe a stage-specific gene that's not expressed in cat stages. But this region up here, whatever this region is, which is in the forward strand, goes from left to right. There isn't a gene model for it, but that region seems to be expressed maybe in all stages. So again, not going to hand wave or anything, but there seems to be something here that's potentially interesting. So we can take a look at these peaks. So the ChIP-seq track and ChIP-chip tracks, which potentially mark transcriptional or promoter regions at the beginnings of genes. And you can see there's a peak right here. And this gene goes from right to left, and this gene goes from left to right. But there's a peak right here in between them. So to me, this would suggest that there's probably something else being expressed right here. And based on the RNA sequence data, which is this blue stuff right here, to me, this would suggest that there is probably something being expressed from left to right here in between these two genes. So that would be potentially super interesting to explore further. Let's see, what else can we look at? So looking down here, we have the RNA sequence evidence track. So I'm going to go ahead and expand this and look at the normal view again. And so now what you can see is, here's the official gene model. These are the introns that match the annotation. And you can see here's an intron that has 4,790 reads that matches annotation. If you click on it, it'll tell you some information about it. Here are the reads again. One interesting metric that we provide is this percent of most abundant intron, or MAI. And basically what we're doing is we find, for this gene, we find the intron that has the most number of reads, and that's considered 100%. And so now let me close this. If we go back here, this one is much darker. This is probably, so this has 6,000 reads, this has 4,000 reads. So this one is probably the 100%. And so now if we go to this one, you see that this particular intron has 6,129 reads, and it is 100%. So it's the most abundant intron, and it's considered 100%. So now everybody else will be a percentage of this 100 of this particular intron, right? So now this one right here is 90%, and then if we go back down to the other ones, this one right here is going to be much lower, so that's only 30% of the most abundant. So then this starts to give you an idea about if you're trying to determine which one of these introns, which one of the gene models is potentially most most abundant. If there are multiple gene models, this could be a useful metric, and you'll see how that's useful in the next thing. Now, if we look down at these introns that unannotated introns with strong evidence, and you'll see here there are multiple. In particular, I'm going to point your attention to this one, which has 5,192 reads. And so this one is actually almost 80% of the most abundant intron. And so if I were to go in and determine whether there's an alternative gene model for this gene, I would determine that potentially this gene has a version where this exon here in the middle is skipped. And I would say that this may be the more dominant version of this gene that is being expressed, basically. Jesse, go ahead.
Yeah, I just wanted to make a comment too about the difference between a gene model and a transcript model, and the fact that, you know, when you annotate a genome, as a rule, we still tend to try and annotate the gene. And Uli should jump in here if she wants to, but the common practice is to annotate the most complete gene with all of its parts. You know, even though in some transcripts this exon is missing, or in some other transcripts that exon is missing, you know, it was all easy in the old days when there was one gene that made one transcript that made one protein, and that's just gone, you know, it's out the window. And so now we have a situation in which you want to try and indicate along the chromosome all regions that have been observed to be part of some transcript family, i.e. products of this gene, but to realize that the gene model that you're looking at may not represent the most abundant transcript, the only transcript. You know, it's typical for human genes to have 30 and 40 different variant transcripts that skip various exons. And we didn't think that happened in the protists until we really began the deep sequencing, and then we began to find it. And so I think it's just important to realize it's not necessarily a question of the annotation being wrong. I mean, in this case, yes, there seems to be a gene missing and the models can be more complete, but it's also a change in our realizations and understandings that the model will usually be the most comprehensive, the largest collection of units ever seen in transcripts, and that there will be a variety of transcripts that one might need to expect. So I just wanted to throw that in there.
Yeah, good point. Uli, was there anything else you wanted to add, or you're happy with that comment? Okay. So the other thing that looking at this gene and looking at a transcription start site, you'll see that there's a transcription start site right here that's going, and it's right here in this section right here, and it's going from left to right. These are really small to see. It'd be nice if we could figure out a way to make these a bit bigger to see. But in any case, you'll see that there's a transcription start site for this gene that is right up here. So technically, if you can take a look now at the RNA sequence data, in addition to this transcription start site, you might be able to extend this gene model or the transcript model a little bit further. And so maybe you can actually annotate the 5' UTR of this gene. And if we scroll up here, and look at the actual RNA-seq data, you'll see that, oh, this is actually quite beautiful here. It actually, the RNA-seq data extends beyond this this genome. Whoops, I clicked a mistake. It has a pretty sharp drop, which I think typically you see sort of a more gradual drop when it's a UTR. Uli, maybe you can correct me if I'm wrong, but it looks like a pretty clean drop right here. So now if we scroll down and look at the long read data, it's a little bit messy, obviously, but most of the transcripts end at the same place where, or maybe start, let's say, at the same place where that RNA sequence data was right here. So so that's pretty cool. So you have three different technologies. You have the long read RNA-seq data, the short read RNA-seq data, and the 5' transcription start site data, all indicating that the transcription start is up here. And so if we go down and look at the community annotation, lo and behold, probably Uli did this, but you'll see that this gene model has already been annotated with a UTR, which extends all the way to the beginning of this transcript. So that's pretty cool, right? And we will, in about 10 minutes or so, Uli is going to give us a short tour of Apollo, which is a tool we have in place that allows you to go in and make these changes, and you can contribute your annotation just like anybody else does to the community annotation, so other people can see. Actually, I'm looking at the Craig annotation here. Let's see where the community annotation is. It's not in there, so you can do it. So this is a gene model that you can annotate in the next section. Some of you may be interested. Maybe Eve, people in your group, somebody in your group may be interested in using Apollo to extend this gene model. So maybe I'll add one more thing. So we have evidence from the transcription start site. We have evidence from the short read data. We have evidence from the long read data. And we have this alternative gene model finder, which extends the UTR to this end here. So this could be a possible gene model. Uli may have some reasons why this should not be annotated this way, and it'll obviously will always be nice to hear those. She has a lot of experience, which is quite nice to have. Okay, anything else, Eve, about this gene model that you thought would be interesting to look at?
I think that was great. Thank you very much, Mar. I
Sure.
thought I saw somebody raising a hand to ask a question, but maybe not. Okay, thank you.
Sure. Let's see, any other group? We have a few more minutes before we switch to the Apollo gene annotation. Did anybody else have a gene model that was interesting? Juan, go ahead.
Yeah. I did have a question. When you were looking at the read data for that specific gene, some of the reads looked like they were independent from each other. They didn't have lines cutting across. I was wondering if they were actual independent reads, and that would lead us to think that there's some sort of two separate transcripts happening, and not necessarily that one model needed to be extended.
Oh, I see. Looking at these reads right here, or which reads were you?
Yep, those that you're looking at right there. Some of them don't have that line cutting across, which I think means that the alignment split them into the gene, right?
Correct.
Correct. I was wondering if they were independent, and all of those that are really neatly aligned would mean that that is a separate transcript altogether.
Right. So it could mean that. It could also mean that your long read data is also sequencing some intermediate species, RNA species as well, that are not fully processed. And so that could be a possibility. And there may be some other nuances of long read data that could happen here. Maybe Catherine would care to comment. But I think, one thing that I think there's one thing you can conclude from this data is that the majority of the transcripts all start at a particular location, right? So we can be confident about that at least.
So I can comment a bit here. So this particular data set is actually very 3' biased. So you often can't draw good conclusions about the 5' end of your transcript from it, but you can draw reasonable conclusions about the 3' end of your transcript. So what I think is happening at the 5' end is this was poly A selected RNA, and I'm wondering if there's a poly A tract in that first intron. And if that's the case,
Oh, interesting.
what could be happening is this stuff at the 5' end is actually mispriming events from a poly A sequence in that intron.
Because it looks, I was going to say this is somehow biased to the 5' end because it looks so beautiful.
Yeah. Yeah, that's really unusual for this data set. It's normally the 3' end that looks good and the 5' end that looks really messy. So I think that may be what's happened here. If that's the case, it can actually be helpful because it does tell you stuff about the 5' end, but that gap in the middle, I don't really trust.
So, and one thing, we can say, well, can we actually take a look at this? And so we do have a track called the reference sequence track, which includes the nucleotide sequence. So, I mean, it might take us a while or too long to actually do it right here, but you can zoom in to this region specifically until you are able to zoom in enough to see what the data looks like. Let's see if this will work. I'll just do like one attempt of this, and if it doesn't work, we can ignore it for some other time. So they are. I mean, I guess I don't know how long of a poly A track you need.
Yeah, I don't know either.
But yeah, you
I also can't
the,
see there exactly where those transcripts are starting.
Yeah, exactly. So anyway, so that's. But obviously, what Catherine is highlighting, an important aspect here, I can see actually there are lots of A's and T's here. So it's possible that this is actually just a highly AT-rich region that is doing exactly what you said. And so the cool thing about this, obviously, is that it's important to understand what the data, where is it coming from, and what the technology, what are the pitfalls of the technology, and also knowing that you can access the various the sequence even of the data and maybe take a look at it this way. So it is interesting. Wherever there's a, so that's probably exactly what's happening here, Catherine, as you described. So it's pretty cool.
Good to know.
Yeah. That said, as Catherine indicated, that this could actually work to our advantage in this case, specifically for looking at the 5' end, of the gene, because it seems that maybe that sort of weird priming that's happening in here allowed the sequence to be much more accurate right here, and so give you many more species that have maybe this is telling us that that's really the correct 5' end of this gene. So. Yeah, but that's really good to know. I always forget about the 3' bias of this data. All right, any other genes people want to talk about, or are we happy to move on to the next section? Okay, I recommend everybody do a stretch, move your arms around a little bit, and get ready for the next section. Okay, I'm going to stop my sharing, and Uli, I'll turn it over to you whenever you're ready.
Okay, thanks very much, Omar. So in the next exercise, it's an optional exercise. We are going to look at Apollo. And you've heard already a little bit from Jesse and from Omar about Apollo. And what Apollo is an extension to JBrowse. So what you've done in JBrowse, you've looked at gene models and at evidence, and then you were able to tell if a gene model was correct or not. What is lacking in JBrowse is, though, the ability to edit those gene models. And this is something that you can do in Apollo. So you can access Apollo from the gene record pages, similar to JBrowse, or you can use the tools menu. And there's this option here to click on Apollo. So for training purposes and to learn how to use Apollo, we've got here a sandbox. And so this is the same site as the normal Apollo site, which you would access from here, but it's for training purposes. So once you've mastered Apollo, you can go to the normal Apollo site but for now, for this exercise, we are going to use the sandbox. So you need to be logged into VEuPathDB to use Apollo. So you use your normal VEuPathDB account. I'm already logged in because I've looked at Apollo before. And so this is now the Apollo interface. And this part is probably already looking very similar to what you've looked in JBrowse. You've got here the same things as in JBrowse, an option to put in a gene ID, can zoom in and out. So what I'm going to do first, when I opened Apollo, I'm going to select the genome that I want to look at, and I'm going to stick with Toxoplasma. So for this practical exercise, I'll walk you through a few examples in Toxoplasma, and I'll show you how to fix gene models. And you will see it's very easy and it's incredibly intuitive. So now we've got Toxoplasma open in Apollo. And what you can see here are these different tabs. And one of those tabs, and this is different to JBrowse, is called annotations. And Apollo is a community annotation tool. And so what you can see here are all of these gene models that were already annotated by the community. So a high number. There are a few left, as we've just seen in the exercise. So Omar pointed out that the gene model we've just looked at in JBrowse could be corrected as well. So the other tab here is the tracks tab. And you can put on additional evidence and also the annotated transcripts. And one important part of this tracks tab is this trackable annotation. So it includes the annotated transcripts. And what you can see here, they look now all of a sudden colored. So this is different to JBrowse, where you just had red and blue, depending on if it's forward or reverse. In this case, in Apollo, these different colors, they indicate the frames. So we've got 3 forward and 3 reverse frames. So what I also want to click on is here in trackable annotation, these introns that match the transcript annotation, and we've already looked at that. So this is similar to JBrowse. And there are also these evidences for introns that were missed with very strong evidence, and this is here. So I also want to put on more tracks, and I know that I like this one here. There is the search option here on top. Alternatively, what you can also do, now that you are familiar with JBrowse, you can use this JBrowse selector and select tracks from here. So this is very, very similar to what you've just used in JBrowse. Okay, but so you can select tracks from here as well. So I'm going to do that, and I'll select those 2. So I've chosen a gene. Let's type it in. So very similar to JBrowse, just type it in or you paste it in. And so here's the gene that I want to show you how to fix. Like before, when we looked at JBrowse, it's quite nice to show the labels. And because it's just a gray line and not as in JBrowse where you can see here these thick red lines or blue lines. So it's always good to put on the labels. So when you look at this gene, what you can see here, that this intron is not supported. Instead, you can see here that these 2 introns, they were not included in the initial annotation. And you can also see that when you look at the transcriptome data, that there is a missing possible exon. So you can add that exon very, very easily. What you need to do is you need to select the gene in the annotated transcripts area by clicking on an intron. Alternatively, you can double click on an exon. Once the gene has a red border, you can just drag it up into the user-created annotations area. Or alternatively, you can use the right-click menu and select Create New Annotation and then Gene. So now, this yellow area, this is different to JBrowse. This is where you can edit gene models, where you can add new gene models and edit existing gene models. So this area is different and not present in JBrowse. So let's add the exon. What you need to do is just select those two introns. With the shift key, you can select two, and then you can just drag them up into the user-created annotations area until you see a green border around the gene. Oh, sorry. Sorry, I clicked by accident back, but no problem at all. Let's go back to the gene. Select. So we're back. Okay, select the two exons and drag them up into the user-created annotations area. Do you see the screen border? And select OK. The next step, what you need to do, once you've dragged up these introns, you need to fill in the exon. And you can do that by selecting those two little exons and use the right-click menu and select Merge. Alternatively, what you can do is select the exon and hover at the edge of the little exon and then drag it up into this area. So the exon has been created, and you can confirm that by clicking on the introns. And once you see here a red border, you can see that they have been considered. So finally, what I want to do is I want to create a 5' UTR. And you can see here that this transcriptome evidence gives you some indication where it can start. What you do, you select the exon and then hover at the edge of the exon and just drag it up until the end. So it's a lot of dragging and dropping. And we are going to do the same for the 3' UTR. Bit too long, but no problem. Okay, so we've created the exon. And there's a particular reason I've chosen this gene. So this gene, when you look at it, it's currently annotated as hypothetical protein. So if you go to the gene record page, which you can do by a right click and then say View Gene Page. So let's have a look at the interpro domains if maybe something was missed and this is not really a hypothetical protein, but there is no data available on the interpro. So there are no domains found in this gene. So if I run interpro for this gene with the additional exon, and you can do that by a right click and then can say get sequence, copy to clipboard, and then you can go to interpro, paste it in and search. Because this can take a little bit, for the sake of time, I've already run it. And you can see now here, just because I've added a little exon, this gene now belongs to a gene family, the selenoproteins. So this just shows how important it is to get a gene structure correct. And what you can do in Apollo is, if you do a right click, there's this menu and you can say open annotation. You can now add the gene product. And we've just said, oh, this is now a selenoprotein. And I'm going to add now the gene product. And it's a type of selenoprotein. I've checked it before how it should be called. And it's based on a sequence model because it's based on an interpro domain. And I'm also adding the interpro domain just so that people know where this came from. So here's the interpro domain. And I'm going to add that here. And just to finalize the annotation, the final step you need to do is open the annotation and select the status finished. And once you do that, and Oma mentioned that already, the next day you can see the new annotation on the gene record page. And I'll just show that quickly, Susan, and then I'll. So as Oma already showed you, and as we've seen in the JBrowse, so just waiting until this loads up to show you where you would see it. And here it is. I had created this on the normal Apollo site, and you can see here that there's now this exon, and we've got here these two introns that were considered in the new gene model. And what is also quite nice, the product description is also shown here in Apollo product descriptions. And of course, this is hugely beneficial for the community because this is searchable. So if you now search for selenoproteins, what comes up, if you click on gene, you can see here that this gene family has been already annotated in Apollo. So you can see here, and this is the one that we've just looked at. This is the Apollo annotations. So anyone interested in this gene family, in the selenoproteins, they search for it, they will now find the gene model that we've just corrected. Sorry, Susan.
Yeah, no, I have a question. Yeah, stay right there. So how do you know that the that's the start site that you want to use and the rest of it is UTR? And you did the same thing at the 3' end where you extended, you saw RNA sequencing reads and extended that into a UTR, but you didn't extend the exon. So I wonder, yeah, start sites make sense.
Yeah, so actually Apollo is great because it does a lot of the job for you. So once you add a UTR, it automatically calculates then the longest open reading frame and the longest methionine then. So this is all automatically done. It also automatically creates then here a stop and the UTR. And what is also different in JBrowse, you don't have to load in a reference sequence. You can just zoom in. And let me just quickly do that to show you that Apollo automatically added here, this here, TGA is a stop codon, and then the UTR starts. So it's really very intuitive because Apollo does a lot of the job for you.
Okay.
Does that answer the question, Susan?
Yes. So I mean, it's based on sequence, right? That Apollo believed that stop site, and so it extended everything on to a UTR instead of, yeah.
And it always goes for the longest here. It might not always be correct, but yeah. Okay, so we've now got the selenoprotein, and one more example before we break. And give you one more example. So similar to JBrowse, that takes a little bit. And whenever you look for a new gene, you have to put on those labels again. And in this case, you might notice that we have these introns with strong evidence. And we have also transcriptome data. Only one track is chosen, but you can add additional tracks. And so this points to a new gene, and you can create it incredibly easy. You just select the introns, and then you track them up into user-created annotations area. Similar to before, you just fill the exons in and do one after each other, and you can see it's really easy and doesn't take long. Do the next one. So we're almost done. And it comes back to the question from Susanne. So now what Apollo does, it looks at the sequence and it looks where is the methionine and then creates the exon and the UTR and sort of here. And the same here for the 3'. Again, it looks at it where the stop is, and then the rest is UTR. And that's it. Incredibly easy, creating new gene models. The last step is just open the annotation and select finished. The next day, it is on a gene record page for everyone to see. Just one last thing to say, and Oma mentioned that already too. So if you're working on a genome, you can of course scroll along and check for incorrect gene models. And one, for instance, is here. Can you see here? Looks as if this should not be an intron, but a very long exon. So you can scroll along. But what you can also do is use the tool that Oma mentioned, the unannotated invent junctions tool. And so this gives you a starter. So you have a list of genes that are probably incorrect, and you can work through those genes first, instead of just scrolling along the genome and correcting genes models like this. And yeah, I think that's all I want to say. And I see that there is another question. Monica.
Yes, actually, I have two questions. One, do annotators look at the user-created models in Apollo in case there might be a mistake? And two, when you zoomed in and looked at the nucleotide level, I have seen some genes that have a question mark, and there is no, there are no nucleotides. So does this mean that there is no sequence available, or that the sequence is? I mean, what exactly does those marks mean?
So I assume this is a gap. Oma, please correct me if I'm wrong with the question mark.
Typically, there are N's and not question marks. So that would be interesting. Which genome were you looking in Apollo, or are you looking in the genome browser?
In Apollo.
Maybe
In
she's
Apollo.
talking about the incorrect splice boundaries, Uli, when they are not correct.
No, it's like a large stretch of just instead of seeing the nucleotides, it's just question marks. I can send you the gene so that you can take a look at it.
Definitely do that. We'll take a look and see if it's possible that some character is not being parsed properly and it's showing up as question marks, or maybe there should be something else. Or so yeah, it'll be very good to see what that is. And then I think to answer your other question, Uli, the first question was about, do annotators check every single annotation for accuracy before it makes it into the official annotation?
Well, typically we get our annotation from GenBank, right? So we can't check every gene.
No, the gene models that the users are providing. So the Apollo. So for example, let's say Monica entered a gene model today, and then in a year from now, we're going to update the Plasmodium or the Toxoplasma genome, and there's Monica's annotation. And the decision is her annotation valid, or do we do anything to validate it?
Yeah, sorry, I've misunderstood the question. So we have some automated checks. So for instance, if you create a gene model that doesn't have a start or a stop, you will get an email the next day telling you that you've got a problematic gene model, and then you can look at it again. We also have monthly emails that you get telling you if anything is incorrect or unfinished in Apollo. And yes, we do look at the gene models that users are providing, but generally, I suppose they're quite good. I mean, Apollo also tells you if something is incorrect, and Gloria already mentioned that. One thing that can happen is that you see this exclamation mark. And so this exclamation mark that points to an incorrect splice site. And in this case, you would just zoom in and then check the splice site and see if it's a valid one. You can also see an exclamation mark for GC splice site, and this is a valid one. But so Apollo already tells you a lot, and also we have these automated checks. Yeah. So they've answered that these question marks, indeed, they are happening in Apollo, and unknown amino acids. You're muted,
What?
Omar.
Yeah, sorry. What is an unknown amino acid in this case?
So if it's a gap, referring to if there are ends.
Oh, I see. I see.
ends,
So it's
the question exclamation mark.
so the question mark is in the amino acid or peptide or world, whereas the ends are in the nucleotide world. So wherever there are ends, then and then the question mark is where the amino acid is uncalled, basically. Okay.
So one thing I also would like to quickly show, 2 more seconds, but the developers worked really hard on making these functional annotations in Apollo searchable. And so I'm doing that now also in ToxoDB. So there is this text search. And if you go there and use a wildcard, and there are these different options that you can search for. And one of the fields is the Apollo annotations. And this is quite nice because it tells you, gives you a list of genes that have an Apollo annotation, a function annotation in Apollo. And so you will see it on the gene record pages, or like I've done before, searching for selenium proteins, those will come up then as well. So it's quite nice that Apollo is connected with our gene record pages. You can see the gene models in the community annotations track, and you can see the function annotation also on the gene record pages.
Great, thank you, Uli. So let me share my screen. And so just so
oh yeah,
you're
sorry,
aware,
I forgot to say about all of that. So we've got an optional Apollo exercise. And so it shows you exactly what you need to do if you want to merge a gene or split a gene. It also shows you how to access the sandbox and the step-by-step guides, what you need to do. And we've got a list of genes, if you're interested in learning how to use Apollo, from different component sites. And you can choose genes from there and then try to fix them in Apollo. And if you go back to the schedule, Omar.
Omar, also reload your schedule because there's another link there that you're not getting.
okay. This one here? No, wait a minute. There we are.
Yeah, so there are screencasts for everything that can happen to a gene. So it shows you how to merge, split, how to shorten a gene, how to get rid of introns, and they match the examples that we are showing in the tutorial. So you can watch it, and because what I've showed now was quite fast, but you can watch these little tutorials and doesn't work.
It takes a while to load sometimes. I think it's a lot of data when you're trying to load it in the view. It's probably might be easier to download them, but yeah. Mm-hmm.
Okay. Yeah, so, and we've got an answer key for the examples that we provide, and that you scroll down, we have quite a number of different genes from different component sites with problems, and you can try and fix them in Apollo. And if there are any questions, we are there to answer them.
Great. Okay, so basically we're at the free time of the workshop. You are obviously welcome to stay on for another half an hour before we close, or welcome to leave. If you're interested in trying out the annotation, I know it's the end of the day, and it's possible that people are just exhausted and you don't want to now go in and try and learn something new. Don't worry about it. The exercises are there and you can try them on your own. The sandbox, just to reiterate, the sandbox that is that is linked in the exercise at the top here, you can do whatever you want there. Nothing you do there will appear anywhere publicly, and you won't disrupt anything. So it's really there for your training. I know Uli said that, but I just want to mention it again and stress that this is really for your training. And so you don't have to worry. I know lots of people worry that, oh, I don't want to change the G model, and then it ends up being wrong, and then I have a mess that I need to deal with. So don't worry. And even if you made a change in the main Apollo, and you think you made a mistake and you're just not sure how to get out of it, don't worry, just send us an email and let us know what you did and how we can help you fix it, or maybe we can go in and fix it for you. So that's, you know, nothing should be a dead end where you feel like you've just done a bunch of work that is you know, cannot be reversed. And so definitely keep us informed about any problems you're running into and any help we can offer. So yeah, so I think a bunch of us will stick on for another 20 minutes and see what questions people have. Feel free to ask questions about Apollo and try out the exercise if you want. Feel free to leave, of course, if you're done for the day, and we'll see you tomorrow. And feel free to ask additional questions if you want. And if need be, we can break out into separate rooms if there are multiple discussions going on. And maybe just by way of introduction for tomorrow, we will focus on a bunch of the functional transcriptomic searches and proteomic searches in the databases. So running different searches, and again, exercising search strategies. We do have a couple of lectures looking at how you can analyze your own data in VEuPathDB, including an introduction to Galaxy, and how you can integrate your data into Galaxy and into VEuPathDB. We'll also have a nice, we'll have an introduction to single cell RNA-seq data. It's a new data type that we just loaded or made available through VEuPathDB. And so Catherine will let us know a little bit about single cell and the program, the application that we use called Cell by Gene, and how you can use Cell by Gene to explore underlying data. So that could be nice. And then we'll talk about orthology and how you can use orthology, leverage orthology in the database for different searches. We already talked about this on the first day, but tomorrow we'll exercise it some more. All right, see you tomorrow, everyone. Bye-bye. Great, thank you everybody for sticking with the workshop, and we'll see you all tomorrow.
Thanks for your question, Susan.
Yeah, sure. It's an honest question. And I noticed on that last one that you did, it really did change that exon. I mean, it looked for the start site, the stop site. I mean, your exon was only like, you know, I don't know, 8 base pairs long or something like that, and you extended that UTR like, I don't know, 100 or something, and it chose half of that to go to the exon.
Yeah, it was a good question. Yeah, I should have mentioned that. Anyway, yeah.
Yeah, that's no big. You know what might go in that exercise is like a table of what sequences to look for splice sites and all that kind of stuff. Might be helpful.
Yeah, I'm actually working on a small tutorial what you need to look out for to see if your gene model is correct or not. And Omar, maybe we can change this. Currently it says, I think, Apollo. Maybe we can change that to annotation. Then I can fit that exercise in there as well. Or not exercise, like a little tutorial.
Yeah, you know, Eve, I have your problem now trying to get to our YouTube channel, even on my even on an incognito window, I can't get into it. It's making me go back and it's just weird. So I can't remember what I need someone who can actually get to the YouTube channel, I think, to make me like I log in as I want to log into YouTube as VEuPathDB at gmail.com.
Yeah, let's take that offline and we'll
Oh,
see
I'm
if we
sorry.
can figure it out. Yeah. All right, I know we only have a few people left on. Is anybody actually trying anything, or are people sort of doing other stuff? Because we can go ahead and close out the session if there aren't going to be any questions, which is totally fine.
I am actually trying, so
okay,
maybe
perfect. Then we'll stick around. We'll stick around. Yeah, definitely. Thank you, Rohit.
Also today I posted one query through that contact us. So
I saw that. Yes, I started. I didn't get a chance to look into it in detail, but I think actually, let me take a look at it right now. So yeah, so you're asking about the status of a gene, whether it's essential or not, and so we don't necessarily make calls based on the high throughput mutagenesis data. So, for example, you can get a list of all the genes, let's say in ToxoDB, that have and their CRISPR scores, for example, but we don't make a call for you and say, oh, look, this is essential, this is not essential, because that's in many cases it's really very subjective, right? So the best we can offer is that we do have the graph that was presented in the paper, and maybe the best to do this by sharing my screen here. Hold on, let me do this. So I'll share my screen, and let's go to ToxoDB. And so, for example, if I ran a search for the CRISPR data, CRISPR phenotype, you'll see that there's a score range here. It's from -6.89 to 2.96. This is basically, let me make sure this is, yeah, this is basically the values we got from the publication, from the people who provided this annotation. And if you scroll down to the bottom of this in the description section, you will see the range of these scores. So this is a graph showing each dot on the graph is one of the genes in the genome, and the score, the y-axis is the phenotype score, so corresponding to these scores right here, -6.8 to 2.96, so close to 3, and certain hallmark genes are marked throughout this graph. And in the paper, they described what they considered as essential, quote unquote, essential versus dispensable, right? And remember, this is based on this particular stage of the parasite. In this case, they did tachyzoids and this particular strain of the parasite. So it's possible that some of these, like VEuP1, is considered dispensable in tachyzoids, but maybe it's really essential in bradyzoids or cat stages. So of course, that's something you have to take into account. So now when you run the search, let's say we can just return all the genes with that had a phenotype score. So we have 8,151 genes. That's pretty much the entire genome. And you'll see here that on the one of the columns that's returned is the CRISPR phenotype score along with the p-value, the standard deviation and p-value. So you can download any of the data. And so, for example, if I go to the download section right here, and I click on tab delimited, you'll notice because this is a search that came back with a column for the score, it's already selected for you. And then you can select any of these columns and then return. So this is going to give you a table that includes a gene ID and the phenotype score. And then you can decide, you can sort them and decide that anything that's above, you know, or below -2 or -3 is a quote unquote important for fitness. I wouldn't call it essential. I think essential is a is a very sort of loose, hard to define term. And so typically you would refer to it as something that affects fitness of the parasite in the particular conditions that were tested. Also, the one thing, yeah.
If you can demonstrate the same thing in Plasmodium falciparum.
Yes. So if we go to PlasmoDB, and so if we go to phenotype evidence, you'll notice that there are multiple different types of experiments available, and depending on the organism, of course. So the RMGMDB database, the rodent malaria genetic modified parasites, it is manually curated, and I believe if you run a search here, you can actually add a text word, right? And so you could type the word, I think, essential here, and select all, and run this. And this should return some results, I believe. And that's because this is a manual curated where they actually, somebody went in and they looked in the publication and they said that this gene is essential, right? So for this one here, these are, in this particular database, it returns all the genes and their particular phenotype. And you can also download these. So when you download this, instead of using this particular column, you have to use the pre-configured table. Go ahead.
Someone yesterday told me there was this piggyback insertions.
Yes. Yeah, absolutely. So I'll show you that as well. So I just want to make sure you're aware of this one here. And then when you download this, this will return a table. You have to parse this out. But of course, there's a lot of textual information here, which you might have to parse out if you're interested in getting this kind of annotation. Now for the piggyback data, same thing. I can get a for any list of genes that I have. Let's start over. So we can go to phenotype evidence, and now if we go to this piggyback data right here, and select the search option for it, and now you will see that can scroll down, and here is the data. And again, to learn more about this experiment, I would recommend looking at the papers. You can look at the description page that we have for this data set, which provides a bunch of information, including the difference between this MFS score, which is called the mutagenesis fitness score, versus the MIS score, which is the I think it's called mutagenesis insertion score. Basically because they're using piggyback, and piggyback transposon inserts in particular sequences. You know, not every gene will be equally mutagenizable, I guess that's the right word. And so some genes may get 1 insertion, some genes may get 10 insertions, some genes may get no insertions. And so that's sort of why they have that score as well. But most people will look at this mutagenesis fitness score. So they're looking at that score. So now let's go back here to the search page, and same thing. You know, it's a score starting at -4.094 or something like that. going all the way to about 3 up here. And so genes that are more towards the negative end are considered important for fitness, and genes more towards the positive end are considered dispensable for fitness in the red blood cell stages of the parasites. And so again, it's an arbitrary value. What you pick, it depends on how strict or relaxed you want to be with your results. So, but you can click and drag. So for example, I'm selecting anything from -1.6 all the way down to -4. And then you'll see here that when you select this, almost 4,000 genes are selected. So maybe you decide, well, this is way too much. I'm going to be a bit more stringent, and we can do this. And now you'll see that 3,000 genes met these criteria. You can be more stringent until you feel like you have a number that's a good number. Again, that's a very, very arbitrary decision. Or you can select all and download the scores. But regardless of what you select, when you run this, you're going to get the same thing. You'll get a list of genes. And for these genes, there should be a column for the mutagenesis fitness score, which is right here. And you can download these by going to the tab delimited. And it's probably, is it selected by default? It's not. So let's find it. So yeah, oh, it is selected by default. So here it is. You can download both the mutagenesis index score and also the mutagenesis fitness score, the fitness score selected by default. And then when you get this, you're going to get a list of gene IDs and whatever other columns you have turned on, but including the fitness score on the last column. Does that make sense?
Yeah, I mean, this was really helpful.
Cool. And did you have another question in your email? I can't remember if this was it.
I mean, yeah, I mean, it was the other was it was a feature request in case, because as you said, the cutoff of MFS and MIS is arbitrary. And so I wanted to know if there is a general feeling in the community that does malaria work, that what the essentiality MFS score range should be.
Yeah, for that, I would honestly go back to the paper and sort of try and gauge from the paper what they did. It's a very hard question to answer easily because it really depends on what the score really means, right? So you could imagine a score of -1 maybe for most genes is dispensable, but a score of -1 for a particular gene means that it's actually really important for fitness. So that's a tricky one to answer, I think. So I would recommend looking at the paper and seeing if you can find any information in there about what they suggest. But I bet you there's going to be sort of this. I remember talking to John Adams when we were first integrating this data. There was a lot of uncertainty about that because for good reason, right? It's not an exact experiment where you say, okay, this is the cutoff score. Anything above it is dispensable. Anything below it is not dispensable. It just doesn't work like that, unfortunately. So.
Okay. And coming to this Apollo one, I was trying one of the gene that was of my interest, and like there are some genes which have like 495, 500. RNA-seq data. How do I, principally choose from them? Which one do I need to go forward with for adding exons?
So you're saying if a gene has, you said more than 500 RNA-seq data sets?
Yes, because I'm looking at one gene right now, and when I go to Apollo, there are like 465 transcriptomic data sets.
So I'm quickly sharing my screen. So what is quite, if I have a genome and I don't know which tracks, I usually use a combined track.
Oh, okay.
And that gives you at least at first an indication, and then you can start looking at individual tracks. But for the first part, what I usually look at is the combined track, and this track is also shown on the gene record pages. in a little purpose window, because it's sometimes quite useful. And sorry, Omar.
Yeah, no, that's exactly right. And one thing I would also recommend, if you decide then to look at a specific data set, is to pick one that has strand-specific data. I think that's always useful. And
if you see at the combined track, does it like combine both positive and negative strand data set?
yes, it does.
Okay, because let's say if my gene is on negative strand and I just need to see the combined track of RNA-seq data that's of negative strand. So
I don't think we have it separated by strand, but you can, right, Uli? I believe we don't have like one combined track for reverse and one combined track for forward. We have them all together. And each experiment also has a combined track. So you could pick an experiment, let's say, that has 10 samples or 20 samples, and then turn on the combined track for that experiment. And I believe that then is colored by strand, by orientation, because it's easier to parse when there are less samples. But with many more samples, it's hard. If the colors were in there, it'll be impossible to see. But yeah, we don't have separated by track.
okay, because and this gene is like overlapping with another gene. That's why
I see. So then I would go definitely pick a data set that's strand specific, and then look at that. You don't have to look at many. You can just look at a couple of data sets and look at them and start to get a feel of what the gene region looks like.
Okay. Thank you. That's all.
Monica, I know you have your hand up. Yeah, no problem. Thank you, Rohit, for all the questions. Those were very good.
This is actually a question for Uli, because I have already edited some genes in Apollo, and I always get this monthly reminder that there's a problem with the splice sites, because it says it's a non-canonical splice site. But if I try to change it to what Apollo is suggesting, then the whole sequence of the protein that goes from that part till the end of the protein is changed, and it's very different from what I have seen. These results that I have are just bioinformatic results. I don't have any experimental data, so that's why I didn't want to change the splice sites to what Apollo was suggesting. So that's what I was wondering, whether you would take a look at this, because I mean, I feel it's right, the sequence, the protein sequence, that it's there, but I also know that the splice sites are wrong. So I don't know what should be done in those cases.
Okay, yeah, I'm very happy to have a look, Monica, if you tell me the gene ID, or I can also search. I do also get those emails from other users. But if you can, probably easier, give me the gene ID. Just
it's several of them, actually, because I work on several of them. It's Naegleria fowleri genes. So I can give you the gene, their genes, but it's like 7, 8, something like that. So you want me to paste them in the chat, or?
Yeah, you can do that, or I can give you my email. What would be easier?
Whatever you prefer. It's the same for me.
Okay. Yeah, you can just post it in the chat, and I'll have a look tomorrow morning. And
okay.
And should be able to tell you something in the afternoon.
Okay, so I'll just paste the whole email, just like I got it. Oh, how can I? Is there a way I can just get the? Yes, I think this is a direct message.
Oh, okay. Oh, Naegleria. Oh, yeah.
So there's quite a few of them, and it's just always bothered me, because I'm wondering whether I did it right or wrong, and it was like a year ago. So I don't know if there's additional information, like RNA-seq data. I haven't looked. So just to make sure.
Yeah, sounds good. I'm very happy to have a look, and I wouldn't worry too much about these emails. These are automated emails, and it's just an indication that there could be something wrong with the splice site. There doesn't necessarily have to be incorrect.
Okay, yeah, because I get them every month and I just sort of avoid them and say, okay, I think this is not a problem. But it's been bothering me for over a year. So,
Yeah, sorry. They are automated, so you will automatically get them every month, and just delete them right away before you get annoyed. But I'm
no,
happy.
it's not a problem. It's just like, I still wonder. So,
Yeah, no.
thank you very much. I appreciate it.
Okay, thanks.
Thanks.
I have one last question.
Yes,
Like
go
for
ahead.
instance, if I have an RNA-seq data set and if I want to, do it on command line, like my own personal RNA-seq data set that have not been made public. So is there a way to sort of upload the coverage file, like in a big format in the Apollo browser, and for my gene of interest, and see if the gene annotation is correct or not?
Yes, I believe, and Uli, you can confirm this, but in the genome browser in JBrowse, you can upload bigwig files, and I believe in Apollo that's also implemented or allowed, where you can upload your own bigwig file to visualize your own data. Absolutely.
Okay. Thank you.
And the way to do it, if I'm looking at, let's see if I still have browser window open. So here's a browser. I'm just looking at the JBrowse, but it'll, since Apollo has JBrowse in it, it should work in a similar way. You can click on track, open track file or URL, and then right here you can drag and drop the or just select the file and select the bigwig file, and then it should automatically detect it. You just have to make sure that the bigwig file, the data was aligned to the same version of the genome that is in the database, right? And so and then once you do that, it'll the coverage plot should show up here. Uli, does it work the same way in Apollo?
Yeah,
Yep,
I mean,
okay.
I just checked it. Yes.
Perfect.
Thank you.
Great. Well, it is past the end of the workshop, and I think everybody is tired, and we will all be back here tomorrow morning to continue the workshop. So unless there are any other burning questions, I say we end the workshop. Thank you, Rahit. Thank you, Monica. Thank you, everybody, for the great questions and for attending, and we will see you all tomorrow. All right. Bye-bye, everybody.
Bye. See you tomorrow.
Right.